Hello everyone. Today we will explain the topic which many of you asked about so much. This topic is antiviral drugs. We will divide it into two separate parts in order to make it easy for you to understand. We will specify this first part about general characteristics of viruses and their life cycle. We will mention some of the most important terms and concepts by which you will understand how these drugs work. The problem that all of you have when you study the antiviral drugs or any others is that you study the topic from the middle. That's because pharmacology books don't give much attention to the basic knowledge regarding other sciences. For example, when it comes to the antiviral drugs, you would notice that books authors starts to explain them directly using terms that they think you know well from other subjects and sciences. From here, all the difficulties arise which make you unable to fully comprehend many of the drugs, especially in the chapter of chemotherapy. That's why in this video, we will focus on the general characteristics of the virus and its life cycle, with main focus on the important points regarding our pharmacology. Of course, we won't illustrate virology in depth, however, we will only prepare you for understanding the concepts and the drugs that will be discussed in the following videos. By the way, antiviral drugs is one of the easiest and simplest topics, as you will see soon. You will need to understand this video, then the next ones will be so much easier. We will start with the virus's structure to give you some ideas about its composition. What you see is the ordinary or the classic form of humans' viruses. The most important part of the virus is the middle part, which is the viral genome. This genome can be DNA or it can be RNA, sometimes it is both of them. Its shape may be circular, linear or segmented, however I don't care much about all of this. You only need to know the most important part of the virus is its genome. The genome is always surrounded by a cover of proteins to protect it and it's called coat or capsid. Both are correct. Viral capsid or virus coat. This coat must be present. There is no virus on the face of the earth that doesn't have this protection. The genome must be protected by this coat layer or the capsid. Both of them, coat and capsid, are together called nucleocapsid. There is a third part which many of you neglect when they study. Now we know two components which are genome and capsid. Still, there is a third component which is these scattered parts in the middle. These parts in the middle are enzymes needed by the virus for the process of replication. These enzymes are called genome-associated enzymes, and these enzymes differ from one virus to another. They are not one set. I'll give you some examples naturally, not all of them. For example, among those essential viral enzymes is an important enzyme called protease, another one called integrase, viral polymerase, reverse transcriptase, and many others according to the type of the virus. Regarding these enzymes, some viruses bring these enzymes with them so they could save the time for replication, exactly like a handyman who brings his tools with them. Some other viruses bring some enzymes with them, but they still need other enzymes from the cell, the host cells. They bring some tools with them, but they still need additional tools from the host cell. Now we have three principal components in the structure of any virus: the genome, then the capsid that covers it, then the principal enzymes, which are essential because he, the virus, knows he won't find them inside the cell. The following components are optional. They might be present in some viruses and not the others. What are the following? Many types of viruses have another protein cover from the outside, like this blue one. This blue cover in front of you is called or is formed of phospholipid, very similar to the cell membrane of our own cells, and mostly they are stolen by the virus from the cell membrane of our invaded host cells. You can call it the outer membrane. This phospholipid membrane is inlaid with these projecting proteins that you see here. They are called spikes, and they are composed of glycoproteins used by the virus for attachment of the invaded cells. These glycoprotein spikes differ according to the viruses. They are not the same. They don't have the same structure in all viruses. That's why it's according to the type of the glycoproteins. These glycoproteins, these spikes, are the ones determining which type of the cells in your body is invaded by the virus. The cells with its receptor. The specificity of the virus invading only one type of cells specifically is something called viral tropism. What does it mean? Consider it a matter of specialty. For example, HCV, hepatitis C virus. This virus goes only to the liver. HIV, the AIDS virus. The virus only targets one specific type of cells, which is T helper cells or CD4, and so forth. Each type of viruses is specialized in invading certain type of cells. What determines this? This is determined by these glycoproteins. And these spikes are the target of vaccines developed by the scientists. For example, if a company wants to design a vaccine for a certain type of virus, the vaccine is mostly targeting these spikes because they are specific to each virus. You can see one family of the virus but with so many strains. For example, I'll surprise you and tell you that the HCV. HCV has roughly about 80 strains, 80 types. Those types originate from the minor differences in these glycoprotein spikes. Very minor differences, but they are still differences which give rise to these strains. Influenza virus. How many types are there? So many. Why? The diversity arises from these glycoprotein spikes. Okay, now the space between this membrane, this outer membrane, and this capsid is not air. It's not an empty space. No, it contains another protein, which is this blue one. These blue dots, this protein is called matrix protein. Matrix is known. It's the protein fill in this space. Sometimes they say M1, but why M1? Why does it have a number? Because I have another protein called M2. It is this red that you see. These red parts, which are very similar to ion channels, indeed they are ion channels. It's called proton channel. Or sometimes they give it another name, they call it M2 protein. Viruses use these channels because when they enter the cell, they need to take off this coat. They melt this protein in order to expose its genetic material, thus it uses these ion channels. A hydrogen ion enters through this ion channel. This hydrogen ion is the one responsible of melting what you see, the group of proteins and spikes, so the virus can perform a process called uncoating. I'll tell you about it within minutes. Now let's count what we said. The optional components that could be present in some types of viruses and not the others. I told you this outer membrane or lipid membrane is one of them. The second is this matrix protein. These spikes are number 3 and the fourth is ion channels. They are collectively called the outer envelope. Some viruses own these components, other viruses don't. I do understand that you have a sensible and logical question right now. Since some viruses don't have these components, especially these spikes, then how can they invade the cells? How can they attach to the host cells? Look, the virus that lacks the outer envelope doesn't necessarily lack all these 4 together. It doesn't mean it lacks all components, no. The virus that is described as non-enveloped is formed until here, the capsid. However, these spikes, instead of being studded into the outer membrane, they are fixed to the capsid itself. Spikes become like this. Also, the ion channels, instead of attaching to the outer membrane, they are attached to the capsid. The missing components are only 2. This lipid membrane that you see, the outer membrane and the matrix protein in here. Only these 2 are absent, and in this case, the virus is said to be non-enveloped. If all these components are present, then we say it's an enveloped virus. What we said is the general structure of the virus. The human virus. Needless to say that viruses are such a large world, and you may find that there are more diversity to it than we know. The size of the virus. Viruses are the smallest particles that you could ever interact with in the world of the infectious disease. The size of the virus varies. The smallest virus, human virus, is about 20 nanometer in the diameter. On the other hand, some viruses can be large like the pox virus, and its size could be around 300 nanometer. Naturally, as long as you are using the scale of nanometer, then this creature cannot be seen except by electron microscope. This is the only way to see it because you are using the nanoscale. The term nano always equals electron microscope. Now let's enter the next slide and see the types of viruses that we have. By the way, the headline that I wrote here, I wrote virus classification, or how we divide them. Of course, this term classification is such a broad one. If I am to talk about classification, then we could stay for a day or two talking about it. However, all I want you to know, me as a pharmacologist who wants you to understand the antiviral drugs, I'll only ask you to know what the most important two types of viruses or the most important two categories of viruses that we deal with. They are just two. The first type of them, which is called the RNA virus, its genome is composed of RNA. And the other type is the DNA virus. Its genome is composed of double-stranded DNA. If you are keen on statistics, then most of the viruses we deal with are RNA viruses. They represent the major class of viruses. The minority are DNA viruses. I'll tell you some examples. Then recently, in the past 30 or 40 years, this man you see is an American virology scientist called David Baltimore. In 1971, he discovered a variant of viruses that we didn't know before, or a different species of viruses. You can call it a new species or new variant, it doesn't matter. They are a new family called retroviruses. I'll tell you about it in minutes. These retroviruses are originally RNA virus. Their genome is RNA, however, they have distinct way of replication. They undergo a reverse process or they reverse a certain step. That's why they are called retro. Retro means reverse or the reverse in viruses. Due to his discovery to this family called retroviruses, he was awarded the Nobel Prize after about 4 years just for this discovery. Because we have an important virus called HIV, the AIDS virus, or the virus causing the AIDS disease. HIV belongs to this family called retroviruses. Before we leave this slide, as much as you can, try to remember or memorize examples of the RNA viruses and examples of the DNA viruses. Because it's not good when an examiner asks you whether the HCV hepatitis virus is an RNA or DNA virus and you don't know. This doesn't pass easily. You, for example, be aware that all the viruses infecting the liver, all of them are RNA viruses, except this one, the HBV. Hepatitis B virus. It is a DNA virus. All the types of influenza A, B, and C, all of these types are RNA. Coronavirus that's causing a worldwide panic nowadays. The family of corona are all RNA viruses. The polio, which causes poliomyelitis or children paralysis, is RNA. Rabies, which causes rabies disease that comes from dogs and animal bites, is also an RNA virus. Measles, mumps, and rubella, or the German measles, all of these are RNA. Some examples of DNA viruses are like hepatitis B virus, as I told you, all members of herpes family, all of the 8 types of herpes family, all of them are DNA. Papillomavirus, smallpox, the monkeypox, which is also causing a worldwide alarm right now, and also the adenovirus. These are examples of some viruses that we may deal with in our lessons. Needless to say, when we will explain the drugs, we won't illustrate all of these. However, we will pick the most important ones and explain their drug. In the following slide here, I want you to keep focused and give me all of your attention, because the drawing that we are going to discuss, or this topic, is the most important concept for the next 2 videos, or in the whole topic of antiviral drugs. This drawing that you see, which we call virus life cycle. It's very enough for me that you understand this drawing and the steps that I'm about to explain. Because if you understand them, I bet you that you will never forget about these drugs. It's the passcode. The passcode for understanding the antiviral drugs is this drawing here, and the organized steps that we are going to discuss. First of all, the virus comes here and attaches to the surface of the cell. As we know, every virus is specific for a certain type of host cells. That's why, first of all, the virus enters your body and searches for its target according to its spikes, these spikes which are glycoproteins. If it detects its receptor on the surface of the cell, then it attaches to it. This cell now belongs to it. It is a host cell. Thus, number 1, the virus comes and sticks here on the surface of the cell, welcomed by its receptor. Of course, I won't let any word pass that could arouse some questions in your mind. I'll answer them as soon as they arise. Now you may wonder why a virus would come to the surface of the cell and be received by a receptor. Did God create receptors for these viruses to make us ill? No, of course not. These receptors which receive the virus and attach to it, they are not originally made for the virus. They were not created for the virus. These receptors are very natural receptors present in our body. Say, for some neurotransmitters, hormones, and many others. Receptors serve in very natural functions. However, by chance, the virus can also bind to them. Thus, we call them receptors, okay? Once the virus attaches to its receptors, then it soon performs the next process, which is called endocytosis, and enters the cell. In other words, the virus gains access to the cytoplasm. The virus enters the cytoplasm. The next step performed in no time by the virus is to take off its protein coat, which we discussed in a previous slide. When I drew the virus and I told you the virus is composed of genome surrounded by something called capsid, then an envelope which has this and that and ion channels and so on. All of this must be get rid of by the virus immediately. It melts them. Why? Because it must expose its genome so its genome can enter inside the nucleus to be capable of replication. This process is called uncoating, and I told you a while ago that the one responsible for this process is the ion channel called M2 protein, or the proton channel. Anyway, the virus takes off its coat or the protein, melting it, then the genome or the exposed genome or the naked genome enters directly to the nucleus of the host cell. Once it enters the nucleus, it will shake hands with your genome. After the greeting and some cups of tea, it performs a huge process called the process of replication or the copying process. The term replication means the virus copies itself millions of copies. In other words, the virus could keep replicating, resulting in millions of copies each 1 hour. The process of copying an exact copy of the genome or the genetic material is called replication. The term replication is a general one in biology. What's replication? Replication is to make copies of the genetic material, whatever the genetic material is. Okay? Now let's explain this replication in detail because it's a vital concept. After the genome enters the nucleus, it copies 2 types of the genetic material. And stay very focused, as I know that the following explanation is not easy to be found at any source, and this is where most of the students get confused during studying virology. Be aware that any virus, once it enters the nucleus, after shaking hands with the DNA, drinking its tea, it copies 2 types of the genetic material. Number 1, it copies an original copy of itself, a copy that's untouched. Look at its genome. It copies an exact copy of itself without any deletion or addition at all. This is the viral genome, okay? After copying a copy of its genome, is this how the story ends? Haven't we just said that the virus is composed of genome, many proteins, enzymes and others? Now, from where does this virus get these proteins? It must copy something else, another genetic material that's called messenger RNA. The recently copied messenger RNA is the one that's sent outside in the cytoplasm. It goes to the ribosome of our own cells and orders the ribosome to translate the messenger RNA in order to synthesize whatever it desires of proteins. How many proteins? God only knows. It's according to the virus. Some may need 6 proteins, others need 10, 20 or whatever. It sends a messenger RNA ordering the ribosome to synthesize the proteins it desires. Which are the components of the virus? Okay, now after sending the messenger RNA ordering the ribosome to synthesize some proteins, what is the ribosome's stand on this issue? It's my ribosome. It should only translate my mRNA and synthesizes my own proteins and no other. In this situation, the virus tells it to shut down, to stop any process of synthesizing host cells' proteins, and only focuses on synthesizing the proteins that I, the virus, need. Thus, the virus shuts down the synthetic machinery or the normal machinery and employs the whole cell for making its own proteins. Now the virus has made two types of the genetic material, as you can see. It made a copy of its whole genome, a copy which is identical, and made a messenger RNA, a copy that's sent with orders for synthesis of its desired proteins. Now I have a group of questions. Number 1. For the virus to be able to copy these two types of genetic materials, either genome or messenger RNA, what enzymes does it need? Or how many enzymes? The virus needs so many enzymes to complete these two processes. Each virus is according to its nature. Say, if this virus is an RNA virus, then it must need an enzyme called RNA polymerase. Polymerase means it is a copier enzyme. If it is a DNA virus, then it needs a DNA polymerase and it might need another enzyme called integrase or reverse transcriptase, and it might need others. What matters is that for the virus to be able to do these processes, it needs many enzymes. I want you to note something. Two things actually. They are very important and I'll help you to note them. The first one. Now the virus needs, say, 6 types of proteins or 5 types or 10 types. Does this mean the virus should make 5 or 6 types of the messenger RNA? Considering that each messenger RNA encodes for a type of protein? The answer is no. The virus only makes one messenger RNA. This messenger RNA goes to the ribosome and tell it, I am the messenger RNA. I am assigned to synthesize all of the proteins needed by the virus. So the ribosome translates the messenger RNA to form all the proteins needed by the virus. However, they get out of the ribosome as one continuous unit attached to each other, because it's only a single strand of the messenger RNA encoding, for example, 10 types of proteins, although it's a single strand. Thus, the ribosome translates it and encodes these proteins, however they get out attached to each other like this. A single strand which is called polyprotein. Polyprotein means a large protein. Now the virus have to divide or cut this large unit as if it were a piece of cloth. Consider it to be a large piece of cloth after it's produced by the ribosome. Thus, the virus uses another important enzyme that's called protease enzyme. This protease enzyme is just like a tailor, and this protease enzyme is brought by the virus. The virus brought it from the start, so it doesn't waste time looking for it. It brings it with it. This tailor called the protease comes to this polyprotein, holds his scissors, and cuts the protein in the determined points. If we are about to make 10 proteins, it holds its scissors and cuts out 10 proteins. And it knows where to cut. This enzyme can never blunder, nor cuts in the middle. This would be a problem. No, each protease knows exactly where to cut. Now I have a question for you. Do you think, for example, this protease is one distinct enzyme in all of the viruses? Allow me to answer for you. Of course not. Each virus has its specific protease. Why? Some viruses, its protease knows it needs to cut 10 proteins in specific points. In other viruses, it needs to cut 6, according to the type of the virus. Exactly like how each president has its own specific tailor who knows his body measurements and how to make the suits fit. So, when it comes to this protease enzyme, we are speaking generally. However, it's not one enzyme. This is according to the type of the virus. Each virus has its specific protease. The second question. You already know that for the virus to be able to make a copy from a messenger RNA, it uses something called RNA polymerase. Do you think that this RNA polymerase is the same in all viruses? The answer is also no, not at all. Why? Because each virus also uses certain type of the RNA polymerase. If all viruses used a single enzyme, or if it were only one enzyme from the hundreds of enzymes that are shared by all viruses, then we wouldn't need a science called virology. For instance, We would design a drug to inhibit this enzyme and it would affect all types of viruses. However, the problem is that each virus uses a group of enzymes. Even if they have similar names, they are still different from one virus to another. That's why you shouldn't tell me, look, when I'll be explaining the drugs, I'll tell you that this certain drug inhibits the RNA polymerase in the HIV virus. You would then think I can use it for inhibition of another RNA polymerase in, say, HCV. No, that's not possible. This polymerase here is different from this polymerase there. Same thing, when I tell you that I have a drug inhibiting the protease enzyme, then it inhibits the protease of this certain virus. However, the same drug won't inhibit the protease enzyme of another virus, and this is where the confusion arises. Anyway, we stopped at this step, which is the tailor called protease, holds the protein, cuts it, trims it, and makes out of it the proteins that are needed by the virus, according to their number. These newly formed proteins, which are extracted and processed by this protease, are divided into two groups. One of them is called structural proteins, which means skeletal proteins, the proteins that form the structure of the virus itself. And there is a type of proteins called non-structural proteins, which are symbolized by the letters N and S. NS means non-structural. Proteins which don't share in the structure of the virus. Then what is their benefit? They are enzymes needed by the virus. Does this enzyme share in the structure of the virus? Not at all. This enzyme is a soluble protein used by the virus, however it doesn't share in its formation. It doesn't share in the structure of the capsid nor the envelope. No. Then these produced proteins, some of them are called structural and some are non-structural. From now on, if I ever mention the term non-structural protein, then I mean an enzyme, soluble protein from these ones, however it doesn't share in the structure of the virus, so you can call it enzyme or something used by the virus. I now have a full genome, identical to the original one. The virus copied it and put it aside from the beginning. Then it sent a messenger RNA to synthesize all the proteins it needs, either they are structural or non-structural. Then the next step, which is the assembly line. Each genome comes here to the side and becomes surrounded by the proteins forming a new virus. This process is called assembly or assembly line, and for this day, this process is still not fully understood. We still don't know how can the virus assemble itself in such accuracy. It brings this genome and these proteins, organizes them, forms the capsid and the channels form itself from the beginning. Still a mystery. However, the process itself is called assembly or the assembly line. Then the final step comes. After the virus has formed itself and become an identical copy of the original invading virus, it's now the time for release or budding, breaching the cell membrane and getting out. Many of the viruses undergo this process without the aid of any enzymes, spontaneously. Some other viruses need enzymes from within the cell itself, from you. They use your local enzymes for their budding process or release. And few viruses, to be able to get out, they need a specific enzyme for themselves. It's not present in your body cells, so from where do they get it? They brought it with them from the beginning. It was one of the enzymes that they brought with them in their toolbox, like the handyman, as I told you. For example, the influenza virus or the family of influenza. All of the influenza viruses are characterized by being unable to get out of the cell without the help of an important enzyme called neuraminidase. That's why some scientists thought of targeting this neuraminidase to fight the influenza virus. Anyway, now I explain the life cycle for you in order to get to what I want, which is our drugs, the antiviral drugs. After we discussed this story, number 1, I can now, from the beginning, design drugs to prevent the attachment from the beginning. The drug either sticks to the virus itself, which means attaches to it, or blocks the receptor which is supposed to receive the virus. On one condition, don't you ever forget it. If I am to block this receptor, then this receptor must be unessential for me. However, if it was essential for my body cells, then I cannot touch it, and I can target the virus itself. This is the first step. You can see that some drugs act on the level of attachment, and they are not many. We don't have many drugs that act on the level of attachment. Maybe 1 or 2 drugs treating the HIV, however, they are not many. Next step, The step of uncoating. I can interfere by a drug with the process of taking off the viral coat. Can I target the proton channel by a drug to prevent this process? Yes. We have drugs preventing the uncoating. However, unfortunately, it's only one drug that prevents the uncoating and in only one virus. We will mention it later when we are explaining the individual drugs. However, I am generally explaining for you where I can interfere. I can interfere here using a drug, I can interfere with the uncoating, however, unfortunately, I can do it only in one specific virus. Now let's open our large crate. Let's see our large playground. When it comes to the replication, 99% of drugs that you will deal with are all drugs blocking this step of replication. These drugs either target the genes responsible for copying of the virus itself, or they target the enzyme responsible for copying the messenger RNA. Any enzyme that's responsible for replication can be mentioned here. RNA polymerase, DNA polymerase, integrase, replicase, reverse transcriptase. What matters is that you target a specific enzyme and inhibit it, so you hinder this replication step that the virus cannot replicate or make copies of itself. Again, for the second time, there is an important condition. The virus could be extremely malicious. Some viruses are wicked as could be. While they are replicating themselves, they are using the enzymes of your own cells, our body cells. In this case, I can never touch them. I can only watch them replicate, because I cannot inhibit my own enzymes. When the virus is this wicked and this evil, I cannot touch it. However, if the virus uses a specific enzyme for its own which is brought by the virus in its toolbox, then I'm able to target it and shut it down. Otherwise, I'm incapable. Thus, you have a big chance to fish any enzyme involved in the replication. Okay? Let me ask you a question. If you are attentive, you will answer it. After the messenger RNA was formed and went to the ribosome, here in this translation process, can I interfere here using a drug? Can I inhibit it? If you are attentive, you will be able to answer, or else you will tell me, yes I can. I can interfere here using a drug to inhibit the translation. No, you cannot. I wish you could, but you can't. Because this ribosome is mine. It belongs to my body cells, and its function is to keep translating the messenger RNA, thousands of the messenger RNA of our cells. If you ever hindered the ribosome or the translation by a drug, then you would kill the creature infected with the virus. Its body cells would stop. Thus, you could never touch this translation. You can interfere here, before it, or after it. However, you can't stop this translation. You can't interfere here. Now let's move to the next step. Here is the big treasure. Here we can play. All of Nobel Prizes are here. Did we agree that all types of viruses must form a messenger RNA which is sent to the ribosome for synthesizing all of its proteins, which were attached to one another before they are separated by the tether called protease into organized ones? We can interfere with this step, the processing, and inhibit it by targeting this protease enzyme and blocking it. Thus, after the virus translates or copies its protein, it cannot cut it nor get its individual proteins. Here lies the treasure. All of the recent drugs that we will discuss are acting on this protease enzyme. Again, for the second time, so you don't forget, this protease enzyme is specific for each virus. Don't tell me, doctor, about the drug that causes inhibition of the protease enzyme of the HIV. Can I use it in the HCV? No sir, you cannot. The protease here is different from the one there. Indeed, they are both called proteases, however, they are structurally different. The step that I'm telling you we can inhibit is called the step of processing. Don't forget about it because I repeat it 100 times in the next video. All what I need you to know when I'm saying a drug is acting on the processing step is that you know where exactly is this step in the life cycle and what is its rule, what happens when I hinder it. If you want to appear as a man of culture in front of the examiner and write a well-organized answers, then the step is called post-translational processing. And it's very reasonable that post-translational means after we translate and form a polyprotein, we trim this protein and organize it by the tether. This is called post-translational modification or processing. This is the fifth step that I can interfere with. The sixth step, the one called the assembly, where it rebuilds itself and assembles itself, like a car production line. Can you interfere with it? The answer is no. But why can't we? As I've just told you, this step is not yet fully understood. Scientists still don't know how the virus assembles itself in such accuracy. How can I act on something that I don't understand? Thus, we have no drugs for the assembly. Now let's see the last step, step number 7, the step of body release, after the virus forms itself and is about to get out. Can you interfere by a drug and stop the release of the virus? Yes, I can. Only in one situation that I have just told you about, the virus called influenza virus. I've just told you that it needs a specific enzyme for its own which is not present in our body cells. No, the virus brings it in its toolbox. It's called neuraminidase. This neuraminidase, if you are able to block it by any drug, influenza virus cannot bud from the cell and becomes trapped inside it, incapable of getting out. Lots of students ask us, what's after we trap the virus? Doesn't it accumulate inside the cell? There's nothing ever like this. This doesn't have any meaning at all. If I block this enzyme or prevent the step of release, the invading virus, which is only one copy, dies, and the cell dies soon after. The cell invaded by the virus dies while it contains it. It's over. It cannot spread. The problem of the virus is that one invading copy is able to copy itself nearly 10 to the power of 10 times. I don't know what's the resultant number, but imagine how big it is. This is within each 24 hour. Accordingly, this invading copy each 24 hours, it releases millions or billions of copies. Now before we leave this drawing, you might wonder about the fate of the cell, which releases millions of copies. What happens to it afterwards? It has one of 3 fates. 1. The cell after it copies the virus many times, the cell itself dies, commits suicide, undergoes something called apoptosis. Apoptosis means it's destroyed or killed. This is one possibility. The second possibility, the cell keeps working. Throughout the day, the virus enters the cell, makes millions of copies, but for how long? Until the immune system deals with the virus. In this case, the cell could survive. It might live. This is one possibility. The third possibility, and the worst one, due to all this occurring distraction, the cell is converted from normal cell into cancer cell. If someone asks you during an oral exam this question, what is the fate or the destiny of the virally infected cell? Tell them there are 3 fates, these 3 possibilities. Now we have finished the life cycle, and if you could understand these concepts, everything would be so much smoother for you to understand when we discuss the drugs. When I tell you that we have a drug that prevents the attachment, then you know, prevents the uncoating, then you know what it means. Prevents the replication, then you also know, because you now have an idea about the nature of this system. Now let's move to the next photo. By this drawing I aim to show you how scientists are able to design drugs preventing or inhibiting the process called replication. The very vital step, our large trait, which is called replication. Didn't we agree that any virus after uncoating and reaching the nucleus, it copies itself and makes 2 types of the genetic materials? It makes itself one original copy and one messenger RNA. And this process was termed replication. Okay, what you see on the left is a DNA molecule, the double-stranded DNA. And you know since high school, in your first or second year, that in the DNA, these cross bridges or these base pairs are composed of 4 amine bases. 4 amine bases. We know these bases since high school. The first 2 of them is called adenine and is symbolized by letter A. This T refers to thymine. The G refers to guanine, which comes from guanosine. And C refers to cytosine. These are the 4 basic groups or the 4 amine bases that form the DNA. In the RNA, if we are to make one, we replace this thymine for another one called uracil. Okay? This information is not new for you. The new one, which I want to explain to you and show you how scientists use it to design the drugs preventing the replication, they told us it's very simple. We will design drugs which look like these 4 bases, adenine, thymine, and the other 2. If we are able to design a drug that looks exactly like them, the same structure or a very similar one, the virus is a primitive creature. It's not a developed one. When it copies itself, it will make a mistake. Instead of taking the natural nucleotide, the natural adenine for example, or the other natural bases, While it copies itself, if you give it a drug similar to the natural one, and as long as you say natural, then you mean analog. The term analog means the look-alike. If you give the virus any drug that's similar to these four, the virus during copying itself and replicating its genome or its RNA, it will make a mistake. And instead of taking the natural nucleotide or nucleoside, it takes the drug instead. Here is a clear example in front of you. This colored one in front of you is the adenine. This colored molecule which I outlined is the adenine. This part attaching to it is a sugar molecule, ribose sugar or pentose sugar. Sugar in addition to adenine are called nucleoside. Look what did the scientists do. They brought the same adenine. Look, it's exactly the same adenine, the same molecule. However, instead of the sugar molecule, they added another thing, a different side chain. Thus it became a drug called acyclovir, the most well-known drug for treating or fighting the family of herpes virus, the 8th type of herpes. The same principle applies to our armory of drugs that could reach 80, 90 or even more drugs. They all act by the same principle or the same theory, which is to design a drug that looks exactly like the nitrogen bases in the DNA. Thus the polymerase of the virus blunders. Here is a drawing from your textbook to make these concepts more comprehensible. What you see, this blue molecule, which is lobulated, is the enzyme called polymerase enzyme. Either it is a DNA or RNA, which is the enzyme used by the virus to make copies of itself. And this is the DNA molecule during its synthesizing process. This enzyme is now synthesizing a DNA or an RNA accordingly. Look how the book illustrates this. This is the natural nucleotide, adenine for instance, or guanosine or thymine. This is the natural. And this in the orange color is the drug which I designed to be its look-alike. Thus, the enzyme, being unevolved unlike our body cells, makes a mistake, and instead of taking the natural nucleotide and starts building it, takes the drug. It gets confused, as the drug is similar to the nucleotide, so it takes the drug and incorporates it inside the DNA of the virus or the RNA. The outcome is the resulting RNA has become corrupted. It cannot be translated nor functions, it's broken. This is the theory upon which is based an armory, no less than 80 or 90 drugs. All of them are called nucleoside analogs. So, from now on, when I tell you that this drug is a nucleoside analog, then you know how it does work. That's it. You now understand that nucleoside analog means a drug that's similar to these four, and it goes to the viral RNA, deceiving it and enters the structure of the DNA or the RNA as a replacement for the natural nucleotide. In the following slide, I want to explain to you or shed some light on the topic of viral replication, because it's a vital topic. We agree that we have a large armory of drugs that acts on replication. That's why I want to explain to you some of the terms and open some topics so we don't need to explain them later with each drug, but only once. I told you that we have 3 types of viruses at the beginning of this video. The first one is the RNA virus. We agree that there is a large class called RNA viruses. The second type is viruses with DNA or DNA viruses. The third type, which was discovered by David Baltimore, who won the Nobel Prize, this type or this family is called retroviruses. What you see here is the viruses after they enter our body cells. This red outline is the cell. This is the cell, and this is the nucleus. I want to observe the behavior of each virus, how does it replicate itself specifically, so I can see which is the best step to interfere with by the drug, from top to bottom. This RNA virus is the simplest of the viruses at all. Once it enters the cell, you know that the next step is the uncoating. Its Majesty takes off its cloak, its protein coat melts, and the virus enters directly to the nucleus. This is the viral genome after it gets rid of its protein coat. It melted it in the cytoplasm. We don't have many problems. This is the genome of your own cells and this is the viral RNA. Translation goes on smoothly just as you know since the first or the second year of high school. The RNA replicates itself via a process called transcription by the enzyme called RNA polymerase. The virus makes 2 copies of the genetic materials as we agreed. It makes an identical copy of itself. An identical copy. Then it makes a messenger RNA to order the ribosome to synthesize all the proteins it needs, as we explained in the previous slide. Thus, we have no problems with the RNA virus. All what it takes is an enzyme called RNA polymerase. This is the principal enzyme that the virus needs. Now let's move to the DNA viruses, which represent a smaller class of viruses compared to the RNA. After the virus enters our cell and finishes uncoating, it enters the nucleus. This viral DNA, when it meets your DNA, it plants itself in our own cellular genome of our own cells. And this is the most threatening idea that you could imagine. Again, you will hear it one more time so you may never forget it. DNA viruses, after they enter our body cells, they thrust themselves inside my genome. At which part? At any place. It could thrust itself in a gene which is a vital one, making a huge problem. That's why I told you that some viruses can lead to cancer. Cancer results from here, from the insertion of the DNA virus in our own genome by an enzyme called integrase enzyme. Integrase, as it causes integration. Thus the enzyme is named after the resultant process. The virus that has implanted itself in our own cellular genome is called latent virus. Latent or provirus. Both names are correct. Latent means a dormant virus. It has already planted itself in you. It's over for now. Sometimes they call it provirus. Provirus means a virus that keeps implanting itself forever. It won't get out of the cells. Each time when your immune cells or your immune system, each time it suffers an ailment and is weakened, the virus which implanted itself quickly uses an enzyme called DNA polymerase. Don't say RNA polymerase, it's the DNA virus. Thus for copying itself it needs a DNA polymerase. It uses a DNA polymerase to replicate and make copies of itself, causing problems for you. I'll give you a simple example for some types of these viruses. A moment ago, in the previous slide or the previous photo, when we illustrated the types of viruses, I told you that we have a virus called herpes virus. Herpes virus is an example for DNA viruses, okay? All of us know that when we have a common cold, any common cold, some vesicles or an inflammation occurs in the mucocutaneous junction at the angle of the mouth, and they are very painful. When you ask your mother or any friend, even commoners will tell you that these are cold sores. They assure you it's from the cold. And if you ask a doctor, he will tell you it's a virus called herpes. Herpes simplex is the reason. However, may I ask you, when I have a cold, does a virus come out of the air and causes this? And each time I have a cold, it comes after me? Does it lurk me? Not at all. This herpes virus entered your body once only, maybe 20 years ago, maybe when you were a kid, and thrusted itself in your DNA. It has become implanted in your DNA. Each time you experience a common cold and your immune system becomes sluggish or weaker, the virus uses the DNA polymerase and replicates itself, making millions of copies, which are causing these sores. The same principle applies to all the family of DNA viruses, to the extent that there is a recently published research about 8 months ago. It emphasizes the number. When they analyzed our human genome, they discovered that 8% of our genome, 8% is such a huge number. Our genome is very vast, 8% of which originates from DNA viruses over the course of millions of years. Since God created Adam and then other humans millions of years ago, each virus comes and implants itself like this and never gets out at all. It keeps implanted inside your own genome, and over the time it may be passed down to generations, to the extent that today they assume that 8% of our genome is originally attributed to DNA viruses. Okay? Now let's see the last one, which is the retrovirus. The man who was awarded Nobel Prize in 71, why he won it? He told us that retrovirus is originally a subtype of the RNA virus. However, it's characterized by something strange. It undergoes a process which is not understood at all. To this very day, we don't know why it does it. It's an RNA virus. It could simply enter the cell like this, copy itself, and that's it. But it doesn't. This virus or the family called retroviruses, which are RNA, before the RNA enters the nucleus, while it is still outside in the cytoplasm, it uses a very specific enzyme that was brought from the outside in the toolbox, not from us. The virus brought it inside its toolbox, and it's called reverse transcriptase. In order to make a DNA out of the RNA, why it does this? We don't know why it does this. It undergoes a reverse process. The logic says otherwise. We all learned previously that RNA is the one that's made out of the DNA. This is the natural sequence. However, going through this process backwards, forming a DNA template from an RNA, you need a very specific enzyme called reverse transcriptase. And this was the reason for giving Nobel Prize. This simple discovery. Why do the virus makes all of this? Because its majesty wants to make a DNA, it wants to form a DNA copy, so it can thrust itself like a DNA virus and keeps implanted forever, exactly like the DNA virus. Because if it sufficed with entering as an RNA and copying itself, it wouldn't be able to integrate. That's why. It's admirably determined on integrating and staying inside your cells as long as you may live. This is the family of retroviruses. The term retro originated from this. That's why scientists said since retroviruses are characterized by this additional step, then we can stop this process completely through targeting the enzyme called reverse transcriptase. And it's very obvious where the term reverse transcriptase came from. It means it makes a reverse process, copying a DNA from an RNA. After implanting itself in the genome, with each time your immune system is weakened, God only knows which enzymes will be used by the virus. It might use its own specific set of genes, or it might turn to be very vicious and uses your own enzymes, those of the host cell, like the host cell polymerase, and starts to replicate. It makes a copy of itself, then it makes another reverse copy, which means, after the virus transforms into DNA, nope, when it replicates itself, it returns to its original form. It makes copies identical to its original genome, which is RNA. Then, any RNA from these, as you know from the previous slide, the RNA gets out and goes to the ribosome and orders the ribosome to synthesize his own proteins, the viral proteins. You, ribosome, shut down all of this. All the proteins that you are synthesizing, stop them at once and work only for me. The ribosome obeys the order and starts to synthesize, as we agreed, the polyprotein, which is all of the viral proteins attached to each other. Then the tailor, that's called the protease, uses a scissor to cut and trim all of these proteins, converting them into individual proteins in order to become usable by the virus. Either to build itself and hence they are called structural proteins, or in other enzymatic uses and hence they are called non-structural proteins. This is the process of replication in detail, and I illustrated all three types of viruses. My goal in all of this is to explain these to you, so when I'm discussing the drugs and telling you that this certain drug can act on the integrase enzyme, then you would know the integrase enzyme doesn't exist except in these two types of viruses, which are the DNA virus and the retrovirus. However, don't you ever tell me that this certain drug causes an inhibition of the integrase enzyme in an RNA virus. That's wrong! The RNA doesn't undergo an integration. Integration only occurs in these two. I can target the integrase and I can target the reverse transcriptase, and we mentioned something about the protease. Now we have finished the first video, which is dedicated to explaining all of the important concepts, ideas and notes. I want to conclude by saying that after you know all about the life cycle and the main terms, we have two types of classifications, and books are confused. All pharmacology books in the world are confused how to classify these drugs. A very lot of books approve this classification for the drugs. It aims to classify the drugs on the mechanistic base. Mechanistic classification. What does you mean by mechanistic classification? It means you classify the drugs regarding the stage of the life cycle. I'll explain them like this. For instance, I have drugs that prevent the attachment, and the book goes on illustrating. I have drugs that prevent the uncoating. I also have drugs for the replication, by targeting any important enzyme from these ones we mentioned earlier. I can also interfere with the step of processing, and I can interfere with the step of release. If the book that you are using is illustrating them in this manner or by this classification, then this book is illustrating or classifying these drugs by a mechanistic classification, like this. For example, a headline, inhibitors of attachment, then it starts counting the drugs. 2. Inhibitors of uncoating, and starts counting. However, this method is being abandoned recently because it's not valid anymore in the recent days. It's difficult, somewhat difficult and confusing. The easier method, the more applicable one, the more practical one, and the one that benefits you more, which also happens to be the one used in the exams, is the second method, which is becoming more popular in books recently. Most of the recent books explain the antiviral drugs using the second method that's called clinical classification. Instead of making students confused over inhibitors of attachment and so on, no. It classifies the drugs like this. We will choose virus after virus, and since each virus is specific and its enzymes are specific for it, then I wouldn't get confused. For example, we will choose the HIV, which is the AIDS virus, and start explaining its drugs. Then we move to the herpes and discuss its drugs and so on. We choose its family separately. This is the method that we are going to use in the following video, in which we will be explaining the individual drugs. And to make it even easier for you, I'll combine the two methods together. I'll start by the clinical method. The headline will be like this: drugs or antiviral drugs against the HIV. While I'll be enumerating the drugs, I'll inform you and tell you that this drug interferes with the attachment. This drug interferes with the system for replication, as we are explaining them. See you in the next video.